engagement opportunity with US EPA, Office of Research and Development. And one more seminar after that will be on May 4th, Water Scarcity and Sustainable in the US Broiler Industry. It's going to be delivered by Walter Budgie. He's the head and director of Center of Excellence of Poultry Science, University of Arkansas, Fayetteville. We are considering, we are planning for the fall semester seminar series. So I encourage you, if you have someone that you would like to have for the fall semester, to let us know so that we can plan for that ahead of time. With that, I would like to welcome our speaker for today, Dr. Lisa Dorso. She's a microbiologist at the USDA ARS Service, Lincoln, Nebraska. And I would like to invite Peter Ampim, who invited her to introduce our speaker for today. Peter.
Yes, sir.
Go ahead, please.
Good morning, everybody. So our today's speaker, as Dr. Ferrer said, is Dr. Lisa Dorso. She currently is a research microbiologist with USDA ARS. She got an undergraduate degree in biology from Illinois State University, master's in botany and mycology from Miami University at Oxford, Ohio, and PhD in food safety microbiology from the University of Nebraska at Lincoln. Her area of focus is microbiology of soils and soil amendments. And she is interested in solving applied problems related to how agriculture impacts the environment and human health. She started her scientific work with state and federal public health agencies and served as an emerging infectious disease training fellow at the CDC. Excuse me. So she's currently focused on human public health and animal health unit. That's why the unit she works with USDA ARS, and in the natural resource program area. So with that said, welcome, Dr. Dorso. Take it away.
Thank you, Dr. Ampin. I'm glad to be here today. I'm sorry I can't see y'all. And I want to do a shout out to Josh, who's worked magic here to allow us to flow smoothly into this presentation. So next slide, please. So here's my contact information. Feel free to reach out if there's anything in my talk today that catches your interest, whether it's that you'd like to apply for a federal job and would like some tips on navigating the federal hiring process, or if you would like to collaborate, or if you're interested in starting your own research program, particularly in antibiotic resistance, and would like help getting oriented in a new field, I'm available. So please don't hesitate to reach out. Students especially, don't feel intimidated. You can email me anytime, and I'm happy to hear from you. Next slide, please. So a little bit about my agency, ARS, the Agricultural Research Service. It's the intramural research arm of the USDA. So we have no regulatory capacity. Those are other USDA agencies. The USDA itself is composed of 17 different agencies, and this circle shows an approximate budget. The majority goes to nutrition assistance, some for farm services, but most of the other agencies are represented by the yellow pie piece, and that includes ARS. One other agency I want to mention is, besides ARS, is NIFA, the National Institute of Food and Ag. They're our sister agency. They have competitive grant programs, both on food safety and in antibiotic resistance. Next slide, please. So I'm here in Lincoln, Nebraska. We have ARS stations across the United States, and our agency mission is finding solutions to ag problems that affect Americans' everyday field to table. Next slide, please. So ARS is a really good place to work, and we hire at the bachelor's, master's, and PhD levels. The research associate title here and the example in the middle, that's the ARS equivalent of postdoc, and they pay great. All of the open jobs, whether it's for us or any other agency, are listed at this website. It's usajobs.gov. So a lot of the jobs there are only posted for a few days, especially the sciencey kinds of jobs. So if you're actually actively searching, you'll need to check back often. And then the hiring process is also a little different than for industry. And the application process can be a little overwhelming if you're not familiar with it. So if you're interested, and I have students contact me about this all of the time, if you're interested in applying for a federal job, email me. I'm happy to share what I know about how the process works and how to maximize your chances of your application making it through the administration, that first admin screening stage, which is where most folks get caught. Next slide, please. So I'll start today with a quick mention of some of my pathogen work because it touches occasionally on goats. And then I'll focus the rest of my time on antibiotic resistance. I'm thinking about it, some of my past and current work. Next slide, please. So I started my career, as Dr. Ampton mentioned, in public health, first the Texas Department of Health, then in Atlanta. That's where I first got started working with E. coli 0157H7. And sometimes it's called Shiga toxigenic E. coli 0157 or STEC 0157. So that many names, they're all the same thing. So I built on that public health experience by going back to school and getting my PhD. And then I did a postdoc at a place called the Meat Animal Research Center in Clay Center, Nebraska, working on pre-harvest control of STEC in beef production systems before moving to Lincoln, where I am now. And if you're not familiar with it, pre-harvest means in the live animal. Next slide, please. So during my postdoc, I worked on a project doing surveillance of the STEC 0157 at local, county, and state fairs. There was a goal to test the hygiene hypothesis. That was like, if the animals were being kept cleaner, would that reduce the prevalence of STEC 0157? At the fairs, the animals are generally hand-raised, a lot of times by 4-H participants, and they're meticulously groomed. They're washed and blow-dried, and the contestants are scored on pen cleanliness. So the animals are kept meticulously clean. The results showed, next slide, please. No difference in prevalence. The fair animals, if we look just at cattle here, 12.6% was compared to feedlot animals at that time, 13%. So quick results there. We disproved the hygiene hypothesis. Not to say that hygiene isn't important. Next slide, please. So that early fair work combined with my stint in public health set me up to partner with public health practitioners on STEC outbreaks, since the methods we used for the fairs allowed us to find STEC in manure and environmental samples, which is kind of like finding a needle in a haystack. The target is there at very, very low numbers. Unlike in humans, so when people get sick with STEC, they're shedding at really high numbers. Next slide, please. So this past summer, I was involved in 2 outbreak investigations. One was raw milk. There's usually at least one raw milk every year. And then this one at a goat husbandry camp. In June in Tennessee, the Department of Health was told that a child was hospitalized in Florida with STEC after attending this camp at this farm. 3 days later, they got another notification about a 2-year-old also hospitalized, and that's when they started their investigation. The farm itself is big. It's it has a lot of attractions for families. They have the petting zoo and pony rides, walking trails, splash pads, food vendors, and then they also have these multi-day animal husbandry camps for kids ages 6 to 10, where the children will pick out a baby goat and take care of it. As part of this outbreak, there ended up being 12 cases, 12 primary cases, 2 secondary cases. So 12 as they got infected directly, secondary getting infected from somebody else who was already sick. Next slide, please. So the work we do with this is all on the environmental side of things. We isolated 3 suspects that ended up being a match with the outbreak strain. We often get just very small samples. This was one empty tube. Sorry, the picture is bad. And then a few goat pellets in one and just a small smear of runny stool in the other. The methods we have are nothing proprietary, but they're just different than what the public health people use. And so they find it helpful to partner with us. And it's always useful to have lab confirmation in outbreak investigations. So I have lots of great outbreak stories that I'm not going to share with you today, but this might be another motivation to email me, and at some other time, I can talk to you or your classes about some of the outbreak investigations over the years. So next slide, please. So this is not on the pathogen side, but on the fecal indicator side. I'm interested in evaluating a new method designed to enumerate E. coli without the need for specialized equipment. So no incubator needed. It's currently marketed for public health and water quality testing, but I think it could be useful for on-farm and field applications. I'm looking at this summer. If anyone's already working with fecal indicators and is interested in joining me, please reach out. Next slide. Next slide, please. Okay, antibiotic resistance continues to be a growing global public health crisis. And there's broad support for efforts to reduce the incidence of drug-resistant infections in humans, their pets, and food animals. So one commonly cited source projects that antibiotic resistance will be the cause of 10 million deaths per year globally. Now, I'm used to working with small numbers. This 10 million number is an abstraction, or it was an abstraction for me until the pandemic hit. So I looked up last week, numbers from last week, global deaths from COVID since the start of pandemic, 6.9 million deaths. So that really puts into perspective the potential impact that we're looking at from these drug-resistant infections. Next slide, please. So first, I'm going to go through a few quick notes on terminology. I'll cover antimicrobial, resistance, and environment, not in that order, starting with antimicrobial. So antimicrobial technically covers a wide swath of microbial organisms. However, though antimicrobial resistance, the term is most widely used in, I see it in discussions, in policy, in publishing. On a practical level, the majority of the work is focused on the bacteria. Sometimes the term antibiotic resistance is used. That's also focused on the bacteria, usually talking about bacterial infections. I use the terms interchangeably, but all of my work is focused on bacteria. Next slide, please. As more people move into this space, and we need more people in this space, other microbes are getting more attention. Right now, concern about antifungal resistance in human infections is growing. Next slide, please. With concerns that resistance is being influenced by triazole fungicide use in crops. So crop people, Dr. Ampin, maybe there's opportunities to overlay some antibiotic resistance questions on top of things you might already be doing, or plots that you might already be maintaining. Next slide, please. So that's antimicrobial. Moving to environment. In public health circles, this is what environment means. It means outside of the patient. Next slide, please. But inside of the hospital. So we can call that built. Perfect timing, you can keep this because there's also water, soil, and air. There is some overlap. Animal housing, wastewater treatment plants, but this natural environment, next slide, please, is what I'm going to be talking about for the rest of today. And the third term to define is resistance. There remains an enormous and important difference between clinical resistance and environmental resistance. In clinical settings, human or veterinary, the resistance is linked directly to treatment failure and patient outcomes. Next slide, please. So resistance, it always starts in the clinic with an infectious disease-causing agent. So something that's already bad enough to make somebody sick and land them in the hospital, or make a patient sick if it's an animal, right? But that we expect to be able to cure with drugs. Then you add on top of that resistance, and the result is treatment failure. So that means patients are sick longer, or they die where they once would have survived. The threat is direct in both space and time. So in the hospital, the phenotypic measures of the disease-causing agent are then empirically correlated to treatment success or failure. So what that means is in the hospital, they see how the bacteria grows on a plate, and then with a certain amount of drugs, and then they see if the patient is cured. And then every so often, they make adjustments so that those two parameters are linked. And you end up with a standardized definition of resistance. Next slide, please. In ag and the environment, we use the exact same word, resistance, but it kind of means something different. Most bacteria in the environment don't even make people or animals sick. And even the ones that do, they have many steps to take before they can make somebody sick. And so the relationship there is indirect. In the environmental realm, there's many hurdles you can think of. If you're familiar with, I come from food safety. If you know the food safety principle of multiple hurdles, in the environment, there's multiple hurdles to go before you actually have a patient being sick. So same word, but from a risk perspective, a little different interpretation. Next slide, please. Next slide, please. So in the environmental realm, when people talk about antibiotic resistance, they are measuring one of three things. These three parameters are entwined, and it's difficult to sort out the relationship in applied field settings at farms and in crop fields. So the three things that get measured: drugs, so antibiotic drugs, those are the drugs that we take or that we give to our animals. Antibiotic resistant bacteria. These are mostly harmless, but some are pathogenic and can cause disease. And then the third component that gets measured are antibiotic resistance genes. These are the DNA instructions that code for resistance. I like to think of them like recipes for resistance. You can't see me, I'm making air quotes around recipes for resistance. Like my mom's banana bread recipe, right? So I make my mom's banana bread recipe, you come over to visit and you try it, you like it, you say, oh, that's delicious. Would you mind sharing the recipe with me? I say, sure thing, here you go. And now we both have that recipe. We both have the capacity to make my mom's banana bread. And the relationship between the bacteria and the antibiotic resistance genes are similar in that there's a lot of sharing of these recipes for resistance among the bacteria. Like banana bread, that doesn't necessarily mean the banana bread's being made all the time, but there's the potential there. So let's see. Next slide, please. So the primary concern for many is the transfer of antibiotic resistance from animals to humans via food, water, and the environment. Advocacy efforts focus concern on the transmission from animals to humans. Really, it's bi-directional via the environment. So you don't really see that or get that sense at the public interface yet. But in the higher level strategy meetings and the policy documents that I'm reviewing, we're starting to see an acknowledgement of the bidirectionality. the flow of these antibiotic resistance components. Next slide, please. And next slide, please. So I'm a little bit of my old research, but first I'd like to say thanks to my collaborators, my technician at the time, and the summer students who over the years have helped collect the data. Next slide, please. So we start with wanting, if we start with wanting to reduce antibiotic resistance in animal ag settings, one of the first questions that needs to be answered is, what's the target we want to set for reduction? What does that look like on the ground? How low can we go? Next slide, please. In order to answer that, we first need to know, oh, no, back one. What's normal or natural level of antibiotic resistance? Is it 0? People think 0 sounds like a good number, but soil scientists know that antibiotics come from soil bacteria and can be naturally occurring. However, the details are scarce when it comes to actually quantifying that or looking at what are the mechanisms for transfer. In the next few slides, we're going to look at what we know about normal or natural levels of antibiotic resistance. So next slide, please. You already got a preview. You're probably been sitting on the edge of your seat saying, ooh, a woolly mammoth. How is she going to tie this one in? We know, talking about normal and natural levels of antibiotic resistance, that antibiotic resistance is ancient. Ice cores from the time of the woolly mammoths, next slide, please, contain antibiotic resistance genes to beta-lactam, tetracycline, and glycopeptide antibiotics. These are antibiotics that we use in the clinic today. Next slide, please. We know that antibiotic resistance is found in pristine habitats, like this cave. Next slide, please. That was isolated for over 4 million years. And yet scientists went in and cultured. They grew bacteria resistant to 14 different commercially available antibiotics. Next slide, please. We know that antibiotic resistant bacteria are found on organic, antibiotic-free meat. And this is an interesting one and an important one that relates to what we do on the ag side of things, even if it's not food safety. Because next slide, please. There are a lot of these consumer studies out there. Sometimes they find a difference, sometimes they don't. This one, they didn't find a difference, but it really depends on which target they're measuring and what methods they're using to measure it. Next slide, please. We know that antibiotic resistance is common in all environmental samples. Scientists have gone across the globe collecting soil, collecting water, and in every sample, next slide, please, regardless of whether it's from a human impacted or a pristine site, the bacteria are found that are antibiotic resistant, including bacteria that are multiply drug resistant. Next slide, please. This is the last one. We know that antibiotic resistant bacteria are found in organic farming systems. Even in the absence of any antibiotics, resistance exists and it persists over time. Next slide, please. In this instance, this is work done by Dr. Rothrock at the ARS in Athens, Georgia, in all natural, antibiotic-free, pasture-raised broiler flocks. These are small and very small flocks. And he was looking specifically at pathogens, but you can get the sense, even here, 64% of the listeria were multiply drug resistant, even though it's coming from a system that isn't using and has never used the antibiotics. Next slide, please. So that's a little bit about what we know about the kind of background and baseline levels of antibiotic resistance in the environment. Next slide, please. So knowing antibiotic resistance occurs naturally, the next question was, are there any obvious differences between the agricultural and the non-agricultural systems? Next slide, please. So I happened to be in the right place at the right time and was able to get my cattle fecal sample sequenced using the earliest of the metagenomic sequencing technologies very early on. Next slide, please. And so I was able to use that to address my question, is there a difference? And how we analyze the data now is very different than back then. How we score antibiotic resistance is very different. It was very simplistic back then, but the result that we found is no difference. There are similar types of antibiotic resistance genes in both the agricultural and the non-agricultural sites. However, next slide, please. Instead, the metagenomes grouped as shown here, with our cattle fecal grouping with dog, fish, and human fecals up in that orange box. This diagram shows out of all of the genes in the metagenome, how many, what proportion were associated with resistance. It ranged from close to 4% for the dog down to 0.7% for the Sargasso Sea. So overall, there's a larger, there is a soil one in here, kind of in the middle, a soy leaf one. But in general, there's a larger percentage of the total genome associated with antibiotic resistance in the gastrointestinal associated metagenomes compared to marine and Antarctic samples. And that's not surprising because it's microbially a much more dense habitat. So there's a lot of competition going on. Next slide, please. These early studies, even though they were very simplistic, allowed us to piece together which bacteria were likely carrying the antibiotic resistance genes in our cattle fecal sample, right? So the Bacteroides, that's an old name, but I'll use it. The Bacteroides, they're thin-walled, wimpy, obligate anaerobes that don't survive well in the feedlot. The Clostridia, on the other hand, are tough, thick-walled spore formers that can survive harsh environmental conditions. So I argue that the resistance genes in Clostridia, therefore, are more important in animal and human health than the same genes carried by the Bacteroides, even though the Clostridia are there at a lower number. Now, the public health and regulatory communities over the past couple of years are starting to embrace E. coli as an indicator for antibiotic resistance, for reasons mainly because they have infrastructure already in place for E. coli. However, as it relates to animal and human health and agricultural manures, other taxa might be biologically more relevant. And so that might be an interesting area if there's a young researcher interested in moving in that direction. Next slide, please. A large component of my research revolves around measuring background and baseline levels of antibiotic resistance. It's an idea that's been gaining traction, but it's still hard to implement. You can almost always bet when a researcher goes into an ag system looking for antibiotic resistance, the conclusion they come out with is there is a lot of resistance. That was my air quotes again, a lot of resistance. It's usually very imprecise. So that's a fine message for advocacy efforts, but it really lacks resolution when it comes to determining acceptable levels for reduction or measuring the impact of best management practices. Next slide, please. But the descriptive studies, these are just building blocks. In and of themselves, they have a very focused and localized utility. The larger aim is not about describing the blocks, but next slide, please. What can we do with the blocks? How do the pieces fit together to build something that's useful to our efforts to reduce the impacts of agricultural antibiotic resistance on animal and human health? Next slide, please. How do we use the building blocks to tailor solutions to individual commodities and production systems? And while there's still considerable knowledge gaps in the descriptive studies, and it remains valuable to pursue those, and I'm pursuing them, we're also at a point where we can start making some testable hypotheses and developing new technologies, new interventions based on the biology of the individual production systems. Next slide, please. My current work, yeah. Next slide, please. So my attention lately has been turning more and more to the biology of the soil. The first interface for most animal manures with the environment, natural environment, is via land application of manures. And I think the soil biology perspective, soil biology approaches are increasingly important as we move forward with agricultural antibiotic resistance research. Next slide, please. It's still common to talk about antibiotic resistance selection at the gene level or the bacteria isolate level. Moving forward, it really needs to be approached from a microbial community level. It's clear that taxonomy and phylogeny, who's there, how they're related to each other, are importantly correlated with antibiotic resistant bacteria and antibiotic resistance gene measurement. We also know that the root microbiome is distinct from bulk soil and has bioactive compounds. So how do these elements impact the microbial community and structure and the resulting changes in antibiotic resistant bacteria or antibiotic resistance gene carriage? Is antibiotic resistance associated with biotic or abiotic components of the soil? And then the hypothesis that I have here, just as the gut is a key location for antibiotic resistant bacteria in animals that live above ground, so too will the guts of the micro, meso, and macrofauna be hotspots of resistance in the soil. So here's another area. Are you isolating any of these soil creatures? If so, it would be fairly straightforward to overlay antibiotic resistance component onto that. Reach out to me. I'm happy to train your student, share my resources. I think there's some interesting questions there. Let's see, next slide. I'm also thinking of broader concepts of resistance across agriculture. How does antibiotic resistance relate to pesticide or herbicide resistance? We know that bacteria have a toolbox of sorts for dealing with stress. Can exposure to one class of stressor impact resistance or tolerance to another class of stressor? We have evidence from individual studies that it can, but the larger trends and details remain to be worked out. Next slide, please. I mentioned hypotheses, and this is a broad one that can be applied to provide actionable data for a variety of stakeholders. We talk a lot about the problems of antibiotic resistance, but are still limited in the solutions we can provide to producers or to consumers. By building on the large body of work that's already completed for foodborne bacteria. So again, this idea of overlaying if you're working in any type of food safety or foodborne pathogen work, adding an antimicrobial resistance component to that can really enrich and enhance the results and information that we can provide to our stakeholders. So here again, an idea. I'm happy for collaborators, or I'm happy for you to take the idea and run with it on your own. Yeah, so the last project that I'm going to share with you today is a multi-agency project that involved EPA, FDA, CDC, and a group of us in ARS. It's built on the framework of the National Antimicrobial Resistance Monitoring System, or NARMS. NARMS was started in 1996, collecting antibiotic resistant profiles from human, food, and food animal isolates. If you happen to be interested, all of the NARMS data is available online. All you have to do is Google NARMS now. And there's actually two websites because FDA and CDC each are putting out their own website. Let's see. So as part of this project, so they've been doing the food and food animal human work for a long time. But as the One Health concept is being more widely adopted, the noticeably absent from this NARMS effort was any work in the environment. So this multi-agency group is working for the past couple of years and through the next couple of years on a pilot effort in this space. As far as ARS is concerned, we're a strictly research-based agency. So we're looking out for, our goal is focused, our main stakeholders are the producers and consumers. The others in the group have more of a regulatory framework for how they approach projects. Next slide, please. The group decided that for the environment, they were going to be working with water. They chose water because water provides a physical connection between people and the environment, because water links habitats, because it's a conduit into and out of ag production, and because it's studied at local, regional, and national scales. So there's scalability there. Next slide, please. So the antimicrobial resistance study is going to be overlaid on the EPA National River and Stream Assessment Study. They call it NRSA. It's run every 5 years. The study collects close to 2,000 samples from across the United States. So some of these sites are easily accessible, and others, they have teams that boat in to collect the samples. They collect water and sediment, the physical and chemical parameters, bioparameters like fish and invertebrates, and other stream health measures. And then the antibiotic resistance data will be added to and correlated with these environmental data. So they'll be taking phenotypic and genotypic measures, including phenotypic enumeration, so culture-based enumeration, and genotypic PCR, targeted gene analysis, and metagenomics, and whole genome sequencing. It's a massive and very exciting effort. Next slide, please. I'll share my culture-based work. That's the component that ARS was involved in that we've recently finished up. Conveniently, shortly after we started, which was just when COVID broke, the World Health Organization came out with their tri-cycle protocol aimed at enumeration of ESBL E. coli in epidemiologically and geographically related human, food, and environmental samples. So that's a lot of lingo there. So ESBL E. coli, ESBL stands for Extended Spectrum Beta Lactamase E. coli. Personally, I am not a fan of the superbug moniker. It's way overused. But if you were going to use that term, the ESBL E. coli would definitely qualify. It's a critical human health threat. Let's see, epidemiologically and geographically related. So the tri-cycle, the tri part of the tri-cycle is that they're measuring in three sample types, so environment, animal, and human, all from the same city, all anchored in the same location, so that they can relate them. So it's expected to be a global standard for AMR surveillance. And the idea was we could potentially align with global efforts, the US effort with the global. Next slide, please. So this is what that looks like. You filter 100 mils of water onto the filter, and then you move the filter onto a selective and differential media. In this instance, it's a media called TBX. You put it on the media with and without antibiotics, and then count the colonies. The E. coli are blue. Technically, the suspect E. coli are blue. As part of this procedure, you have to pick 10 colonies and then confirm as E. coli and as ESBL. Next slide, please. So the NARMS group evaluated 3 methods. The WHO tri-cycle method that I already mentioned. Then we wanted to anchor our efforts with a standard method. We chose the EPA surface water method, modified with and without antibiotics. And then we also added the Quantatray CoLalert method. It's a approved standard method for the enumeration of E. coli from surface waters, and it's widely used in water and wastewater communities. It's way quicker to process, especially with turbid samples, something that we thought was going to be important if we were aiming to process 2,000 samples over the course of 2 summers. Next slide, please. We did a comprehensive set of validation studies. One element was a cross-lab evaluation using surface water from 4 areas of the country, evaluated in 5 labs for all 3 methods. Next slide, please. Regardless of the source, wherever it came from, all of the isolates got run through the tri-cycle method for confirming ESBL, which looked like this. So the yellow, kind of yellowish portion of the plate, that's where the bacteria is growing. It was spread over the whole entire plate. Then those white circles are paper discs impregnated with the drug. So those go on as soon as you inoculate the plate, and they diffuse into the media. And then those circles are the zones where the bacteria is not growing because it was inhibited by the drugs. And so those standardized clinical methods that I mentioned at the beginning, they have charts, and you can measure how big that zone of clearing is, and then determine if the bacteria is resistant or sensitive to each particular drug. Next slide, please. We did a separate set of laboratory evaluations to determine the impact of turbidity on filter time, the readability of the Quantatray, and the recovery of E. coli for a range of turbidities from 0 to 1,000. NTU is a Nephil, I can't say it. It's the, I could say it, except right this moment now, it's leaving me. So it's a measure of turbidity that we use to score the, how much particulate matter is in the sample. Next slide, please. So the filtration times range from 22 seconds at 0 turbidity units, 31 seconds at 5 turbidity units, and 30 minutes for the 751. The filtration of the 1,000 NTU sample was halted after 33 minutes in my lab, but one of the collaborators had enough patients to run it the whole time, and it ran for 45 minutes for one sample. Recovery of the spiked E. coli, you can see in the photo there, declined with increasing turbidity, mostly due to that thick layer of sediment that either obscured the colonies or it clogged the filter and pushed all of the colonies to the edge. The turbidity differences were noticeable in the Quantatray, there on the bottom, the Quantatray trays, but it was still possible at both the medium and the higher turbidity levels to discern both the yellow, and then this test you can tell yellow is coliform, and if it's yellow and it glows, it's E. coli. And both of those could be read even at the highest turbidity levels. So that was the screening that we did for the NARMS study. Next slide, please. Is the group of people involved in the NARMS work. The project was led in the middle there. Allison Franklin is a postdoc at EPA. Jay Garland is the EPA lead on the project. FDA lead was Pat McDermott, and ARS lead was Kim Cook. So this is a massive effort, and they are moving forward, starting right now, there's a preliminary watershed component going on, and this summer they'll be starting that national survey. So next slide, please. My almost final slide is a big thank you to my technician, Justine Condon, who did a massive amount of work for these validation studies. Next slide, please. Thank you for your attention. I'm happy to entertain any questions.
Thank you, Dr. Dorso, for this excellent presentation about different topics in a very important area of health. both animal and human. And we open the floor for the questions and answers. I'd like to encourage our colleagues to post their questions in the question and answer section of the webinar, and we'll be happy to entertain them.
While my colleagues are thinking about their questions, Dr. Dorsa, great presentation and really appreciate you. I'm the Executive Associate Director of Research here. My question is, could you comment on the trends with gene-environment interactions in light of the climate change? And can you kind of talk about some mechanisms that might be changing? I know you talked about E. coli primarily, but any comments on that? Thank you.
Yeah, so that's somewhat outside of my research area. So I don't really have any, I have to be careful answering things outside of my own research area in these forums where I represent the federal government. But certainly on the pathogen side of things, we know that climate change is impacting what is able to live where, and there are impacts, but I'm not qualified to answer that question. I'm sorry.
Well, I understand because there have been recent threats of fungi spreading across our country and the globe. And I know that antifungal drugs are far less than antibiotics, but thank you. I understand.
Yeah. Well, the antifungals, hot. There's a lot of funding. I'm part of a group that reviews USDA policy documents on AMR and how we respond also as a country and global policy documents that might impact agriculture. And that has really been a top priority now. And I think that's starting to be reflected also if you look at the NIFA framework for grant proposals.
Great, thank you.
Great. So I think while others are looking for questions, I would like to also ask another question if you don't mind. I think
I'm happy to not be able to answer another question.
No, okay. Well, the question is about, you talked about bacterial resistance. And as a soil scientist, we know that soil plays a major role for potential future antibiotic medicines that could be solve this issue or address this important health issue. Can you comment on that one?
Yeah, that's certainly the case. Classically, soil has been mined, soil bacteria have been mined, and soil fungi and actinomycetes, however you classify them, have been mined for antimicrobial compounds. And then that hit a lull. But there's been a resurgence in that with a lot of the new technologies that allow for high throughput screening and some technologies that are allowing us to culture previously unculturable drugs, pairing that with the metagenomic methods and gene-based analyses that can give us clues to what's there. And we can mine that data and help use that data to target those efforts. There's also some really, I think, productive and useful programs out there geared at students, bringing actual discovery of new antibiotic compounds into the classroom as part of experiential learning opportunities. So kind of on the really big scale and also at the local scale. Some exciting things going on there now.
Great. So I think we don't have questions, but I would like to.
I have a question.
Go ahead, Peter.
Dr. Dorso, you said something about the microbial resistance and you linked that to herbicide resistance. Could you throw a little bit more light on that? Because based on my knowledge on herbicide resistance, you are looking at something describing the mode of action. It could be an enzyme, could be a site, or some other thing, or some biochemical pathway within the plant or the weed. And so how, in your opinion, is bacterial resistance kind of linked to herbicide resistance? Any
Okay.
comments?
Yeah, this is a good one. And I think there's a couple of possible different mechanisms here. First, the resistance elements are the antibiotic resistance elements, for the most part, are exceptionally mobile and moving a lot around in the genome. So anything that provides a selective pressure to the host will also be impacting. So there's hosts on a couple of different levels. If you have bacteria that are plant associated, and then there's a selective pressure applied to the host plant, that's going to provide a selective pressure also then to the bacteria that rely on that plant, whether it's above ground or below ground in the rhizosphere. As part of how the plants, the own plant biology, there's chemicals that come out through the roots, again, that can provide potential selective pressures there. So that's one of the possible ways that I think that it might be related.
Okay, I have maybe a quick statement to make here. We do have some work that we've been doing on bacteria in soil in response to organic amendment types and rates. And we've seen some variability in the fauna there, did some sequencing work. This is an area where we are interested in basically exploring further, and that's maybe opportunity for collaboration. Also, maybe, I'm not sure you're aware that we have a Goat Research Center here. So maybe there is also potential for in working on the aspects that as it relates to animals. And one more comment. Do you know Dr. Tala Awada? She's the Associate Dean for Research. Yes, she's a friend of mine and also colleagues. We collaborated in a few projects. Please tell her my regards for that one.
I will. I'm part of a big project that she's leading here, looking at nitrogen. It's called the Nitrogen Rate Project. It'll be the start of a long-term crop-based research project. So that also has an amendment component to it. So
well,
yeah, I think we should follow up after this.
I agree. Well, we would like to give you a time to close out this webinar. We really appreciate it. And can you please tell us the closing remarks?
Okay, well, thank you very much for inviting me. I appreciate your attention, and please feel free to reach out to me at any time if you have any questions or are looking to collaborate or are looking for collaborators. We have a lot of resources in ARS, and we're very interested in encouraging the next generation of scientists and providing them opportunities. So thank you very much.
You're welcome. Thank you, Dr. Dorso, and really appreciate it. Please give her a virtual round of applause. And you have a great rest of the day. Thank you.